Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon, everyone. The topic for today is the detection of SARS-CoV-2 proteins by Aptamers-based assay. An interesting topic, isn't it? Let's begin with the principle behind each technique. What exactly are aptamers? Aptamers are artificially synthesized oligonucleotides that are capable of specifically binding to a target antigen. Aptamers are usually generated by an in vitro selection and amplification technology called SELEX. Aptamers can bind a wide range of particles in both inorganic and organic particles, including proteins. Aptamer has been shown effectively bind the whole viral particles and even cells. The high specificity, sensitivity, and binding affinity to the target molecule is particularly important property of aptamers in the cited context. Now let's talk about the method or process involved. We have enzyme-linked aptamer binding assay. This aptamer has been used to detect and treat the parasitic infection caused by these parasites in human beings. It is also used as a substitution for antibodies as the bioreceptor or the transducer. This aptamer is also known as an enzyme-linked oligonucleotide absorption test or an enzyme-linked aptasorbent assay. This aptamer detects the influenza A virus, H5N1, and targets the hemagglutinin protein. This sandwich enzyme-like aptasensor recognizes the H1N1 and H3N2 subtypes. Now let's dive into SELEX, systematic evolution of ligands by exponential enrichment. It is often modified to make the selection process more efficient and less time-consuming, and to select aptamers with particular binding features such as affinity and specificity for different target molecules and for different application. The first step of conventional SELEX is to incubate the sequence pool with the target, such as protein or nucleic acid. The random region contains the sequences that will be tested for high specificity and affinity to the target. The second step is when sequences that bind the target is kept, while unbound nucleotides are removed. The third step is to purify and amplify the bound sequences to form a new sequence pool for the next cycle. A negative selection or counter selection step involves incubating the sequence pool with target analogs, undesired subtypes, or the unbound sequences. This step can take place before or after target incubation to improve the specificity of candidates.
In addition, let's go through into detail about the advantages of using aptamers to detect COVID-19. Firstly, small in size, so it able to bind with more recognition molecule. We can say that it has high target potential. Secondly, the DNA aptamers and modified nucleic acid aptamers are chemically and thermally more stable than protein, so it allows them to have longer shelf life. Third, they have high binding affinity and specificity, which is able to recognize the target even in low concentration and ignore other target even in high concentration. Next, easy and controllable modification. Besides that, no intrinsic immune response because chemical use is nucleic acid. Last but not least, it is stable in different environmental conditions. Without wasting any time, let's move to the limitation of this technique. First, technically demanding and costly. Second, RNA or DNA contaminants. Third, risk of excretion by renal filtration. And next, may produce error result. It may be false negative or false positive result caused by low RNA stability. And lastly, the presence of enzyme inhibitors in biological sample. That's all from us. Thank you for watching this video. So now, the floor is open for the question and answer session.